So welcome to the third Academy4 research webinar. Today we'll talk about brain obesity and morphometrics with Clara SANCHEZ. Clara received a master's degree in genetics and development at Université Côte d'Azur in July 2019. She then started a PhD thesis with Dr. Carole Rouvert and Eric Debreuve in the lab of Dr. Jean-Louis Naon at IPMC Institute. Clara is also an assistant lecturer at Université Côte d'Azur, where she teaches biology and genetics to bachelor students. Today, she'll talk about the effect of a sunflower oil-enriched diet on microglial reactivity, neuroinflammation, and cognitive disorders associated with obesity. Clara, it's your turn.
Hi, everyone. Thank you for inviting me to talk about my project. So, first of all, I'm Clara SANCHEZ. I'm a second-year PhD. I'm supervised by Carole Rouvert and Eric Debreuve. And this thesis project just is the following part after my Master 2 internship that was funded by Academy4 at Université Côte d'Azur. And my thesis project is funded by UR Life and DS4H, and is titled The Effect of a Sunflower Oil-Enriched Diet on Microglial Reactivity, Neuroinflammation, and Cognitive Disorders Associated with Obesity. So let me just share my presentation. So let me introduce you that obesity is a major public health problem. It represents 13% of obesity worldwide and 39% of overweight worldwide. It's defined by the World Health Organization as an abnormal or excessive accumulation of fat that can be harmful to health. And so it leads to numerous consequences. The two main are the type 2 diabetes and cardiovascular diseases as stroke. And it also can lead to certain type of cancer and sometimes cognitive disorders. In the body, obesity is associated with a peripheral inflammation, so characterized as a low-grade systemic inflammation and defined as a production of a pro-inflammatory mediators as IL-6, IL-1 beta, TNF-alpha, and the tissue infiltration of immune cells. Recent study also shown that obesity is, associated with a central inflammation, so particularly in the region of the hypothalamus, implicated in the feeding behavior. It's also defined as a production of pro-inflammatory mediators, as IL-6, IL-1, TNF-alpha, and by a glial activation of two glial cell types, which are the microglia and the astrocytes, that change their morphology when they are activated. So just as a definition, these two cell types are microglia and astrocyte. Microglia is the immune cell of the brain, so the central nervous system. It's able to phagocytosis, scavenging, and extracellular signaling and the production of these pro-inflammatory mediators. And astrocytes have many roles as the metabolic support from neurons, the blood-brain barrier maintenance, and the physical structuring of the brain. Here is an example of a study which shows that after 4 weeks of a high-fat diet in the brain, and here particularly in the region of the hypothalamus, you can see here a proliferation and a ramification of the microglia cell that is a witness of their activation. So here are for the consequences of the obesity, but what about the cause? In the body, we have an energy balance, which is defined as the sending of signals from the periphery to the central system. Signals are going to go to the hypothalamus, which is going to regulate the food intake after to have intake and expenditure at regulation. Sometimes, when there is too much intake or not enough expenditure, the energy balance can be deregulated, and this is the cause of obesity. What are the factors that can deregulate this balance? So, principal factors are the sedentarity, medicines, nutrition, genetics, and psychology. But the main factor of obesity is the nutrition factor, because a lipid-enriched diet is going to lead to a lipid storage increase in the adipose tissue of the body and to obesity phenotype. Studies also shown that a lipid-enriched diet can lead to inflammation in the body. Why? There is many types of lipids. So lipid can be fatty acid saturated or unsaturated. It can be mono or polyunsaturated. And inside the polyunsaturated fatty acid, it can be of two types, omega-3 that are anti-inflammatory molecules and omega-6 that are pro-inflammatory molecules. It exists a balance between these two types of molecules in the body because they are in competition for the same enzyme, and so in competition to produce pro and anti-inflammatory molecules. Our hypothesis is that this balance, omega-6 and omega-3, is going to have maybe, an impact here on the inflammation triggered by a lipid-enriched diet, and maybe can modulate the link between obesity and inflammation. To do so, we have fed mice with 3 different diets. So a standard diet composed of 5% of lipid and with a normal omega-6 and omega-3 ratio of 6.5. We then have 2 high-fat diets enriched in 20% of lipid, the soybean corn enriched in polyunsaturated fatty acid to obtain a quite normal ratio of 7.3, and the sunflower high-fat diet really enriched in polyunsaturated fatty acid to obtain a deleterious omega-6 and omega-3 ratio of 21.3. Mice are fed during 20 weeks, and the first analysis we perform is the incorporation of all the lipids contained in the diet in the body of mice. So here is a correlation analysis between here lipid categories contained in the diet we gave to the mice and lipid categories found back in circulating triglyceride after sowing mice. You can see here that between the categories found in the diet and in the circulating triglyceride, we have a really strong correlation, close to 1, meaning that the more we gave, for example, the saturated fatty acid in the diet gave to the mice, the more we found back in the circulating triglyceride. It's quite the same for all the categories. We then perform analysis of correlation for the lipids ratio that is interesting for us. So the omega-6 and omega-3 ratio, the acid linoleic acid and alpha-linolenic acid, which are precursor for the omega-6 and omega-3 ratio, and 2 omega-6 and omega-3 fatty acids that are also important. You can see here that we have a strong correlation again between the diet and the red blood cell membrane for the lipids ratio, meaning that the lipids category are well incorporated in the circulating triglyceride, and the lipids ratio are well found back in the red blood cell membrane. So this allows us to continue our study because the ratio is well conserved, and so we can work on. So our project is separated in 4 parts: the effect of the lipid nature, and so the omega-6 and omega-3 ratio, on first the obesity development, then the peripheral and central inflammation. After, I'm going to talk about associated cognitive disorders, and after, the hypothalamic microglial reactivity, which is a morphologic analysis in collaboration with I3S and my co-supervisor, Eric de Breuve. So first, for the first part, so the obesity development, I'm going to talk about the weight gain. So you can see here on the weight gain curve that both high-fat diets, so the soybean corn and the sunflower diet, is triggering a strong weight gain in comparison to the standard. But they're not different from each other, meaning here that the omega-6 and omega-3 ratio have no impact on the weight gain. We then perform analysis of parameters associated with obesity, as first the glycemia of the mice. We have to measure the blood glucose level, and we have also a difference for both high-fat diet that triggers hypoglycemia in mice, but again, they are not different between the 2 high-fat diets, so not different independently of the omega-6 and omega-3 ratio. Same analysis for the lactinemia of the mice. We perform measurement of the serum leptin, and both high-fat diet triggers obesity with these parameters increased, but not influenced by the omega-6 and omega-3 ratio. The last analysis of this part of obesity development was the measurement of the glucose regulation, so by performing 2 types of tests. The glucose tolerance test, which consists in an injection of glucose first, and then we see the regulation of glycemia during 2 hours, or the opposite, the induction of insulin. And we see the regulation of the glucose level, so the glycemia. Here you can see that both high-fat diet have deregulation in the glucose homeostasis, but again, it's not different between the two omega-6 and omega-3 ratio, meaning here that the ratio have no impact on the obesity development and the obesity phenotype we observe. I'm going to then talk about the peripheral and central inflammation. So I'm going to start with the peripheral inflammation by presenting you the expression of pro-inflammatory markers that are TNF-alpha, IL-1 beta, IL-6, CCL5, and CCL2 in first the SC-WAT, which is a subcutaneous white adipose tissue, so peripheral tissue. You can see here that only the sunflower diet-fed mice show an overexpression of the 4 marker here, pro-inflammatory marker. And it's quite the same in another white adipose tissue, which is the PG-WAT, so the perigonadal white adipose tissue. We have the same pattern of pro-inflammatory markers expressed in the sunflower diet-fed mice. We measure the expression of this marker in the third tissue of the periphery, which is the liver, and we have quite the same pattern. So here for the inflammation in periphery, only the deleterious omega-6 and omega-3 ratio, which is the sunflower diet, increased the expression of this marker. Let's go for the brain. So we measure the inflammation in 2 regions in the brain. First region was the hypothalamus, which is the brain region implicated in the feeding behavior. And the second region was the hippocampus, which is implicated in anxiety and cognitive processes as the memory. As I told you, that obesity is sometimes associated with cognitive disorders. So to begin with the hypothalamus, as the peripheral tissue, we observe that only the sunflower diet-fed mice show an overexpression of this marker here in the hypothalamus. And it's quite the same conclusion for the hippocampus, but only for 3 markers on 5. This is meaning that only here in the brain also, the deleterious omega-6 and omega-3 ratio is going to increase the expression of these pro-inflammatory markers in the 2 brain regions we measure. So the hippocampus, I told you, is implicated in anxiety and cognitive processes as the memory. So it leads us to look at the anxiety behavior and cognitive disorders. So hippocampus is implicated in the memory processes. We first measure then memory in mice. So the task we perform is the Morris-Watomase task, which take place in 5 days. So the first day is the Q task. It's a visual validation of the test to verify that mice have no visual alteration due to the diets. Here, we have no difference in the latency to reach a platform for these mice. There is no visual alteration. We then train mice to find a platform immersed in a pool for 4 days. And you can see that all the groups of mice learn well the spatial localization of the platform because the latency is decreasing in the same manner for all the 3 diets. But the interesting thing here is that at the first day of learning, we perform 4 trials per day per mice. And you can see that for the sunflower diet-fed mice, at the 4th trial of the first day, they totally abandon the looking for the platform. This is something really interesting and showing maybe a behavior of depression or sometimes anxiety-like. After the training and the learning of the platform for 4 days, we take out the platform and we perform what is called a prop test, meaning that we measure the time spent in each quadrant looking for the platform for each mice. So the name of the quadrant are the T for the quadrant where was the platform, opposite, left and right. You can see that standard mice spend 55% of the time looking for the platform in a good quadrant. Soybean corn spend quite 50% of the time, and sunflower diet-fed mice only spend 40% of the time looking for the platform in the good quadrant. So here we have memory deficits for the sunflower after 24 days after learning. It's also interesting because sunflower diet-fed mice made less platform crosses of the precise spatial localization of the platform in the pool, meaning here all these results that the deleterious omega-6 and omega-3 ratio, the sunflower, seems to induce a spatial memory deficit in the Morris-Watomase task. To verify these results and to continue analysis, we perform actimetry tests, so the locomotor activity tests, consisting in putting the mice in a cage with laser to measure its activity during 3 days, composed so of 12 hours light phase and then 12 hours dark phase during 3 days. You can see here in the curve that the sunflower diet fed mice show an increased activity during the 2 first dark phase. Sorry. In comparison to the other high-fat diet, which is the soybean corn and the standard diet. So the sunflower diet fed mice are hyperactive. And this is sometimes something linked to an anxiety-like behavior. So we first measure an hormone in rodents, which is the corticosterone, and it's the principal stress hormone in rodents. And here it is, we have an increased circulating corticosterone levels in mice fed with the sunflower diet. So we continue by measuring the behavioral, the anxiety-like behavior, with 3 behavioral tasks that are the open field, the dark light box, and the zero maze. All these tests are based on the same principle, which is that an anxious mice is going to spend less time in the anxiogenic part of the apparatus, which is the center of the open field, the light box of the dark light box, and the open arm of the zero maze. Here are the results. So you can see here that for the time in center in the open field, sunflowers spend less time in the center, less time also in the open zone of the O maze, and less time in the open zone of the dark light box. And in the dark light box, they also take much time to go through the first exit of the dark box through the light. All these results are showing us that the deleterious and the sunflower diet is triggering an anxiety-like behavior. So all these results showing us that the sunflower diet fed mice are got inflammation in the brain and show cognitive deficits linked to the inflammation of the hippocampus. So to go through the last part of the project, which is the hypothalamic microglial reactivity, we first measure the expression of a microglial marker, which is IBA1, and measure its expression so in the hypothalamus. You can see that as the inflammation, only the sunflower diet fed mice show an overexpression of this marker. So we perform analysis of the morphology because I told you that when microglia is activated, it's going to have proliferation, smaller and more circular soma, so a changing in the morphology, and also increased number and length of extensions. So to measure this, we perform microglial morphology analysis on mouse brain slice in the region of the hypothalamus by doing confocal microscopy and immunochemistry, so of this marker IBA1 to mark the microglia in mouse brain slice. So the only results I have to show you today is the dendrite length and the proliferation of microglia. You can see here that dendrite length seems to be longer for the sunflower diet fed mice in the region of the hypothalamus. And also, it seems that microglia have proliferate in the region of the arc, which is the arcuate nucleus. It's a specific region inside the hypothalamus implicated in feeding behavior. So these results show that the sunflower diet fed mice seems to have a proliferation and a ramification of microglial cells. Let's talk about morphometrics part. So just to explain to you how do I measure the morphology. So here is an example of a brain slice of 30 micron thickness of a mouse brain. Here is the region of interest, so the hypothalamus, and all the green marker is the IBA1 marker. Here is a zoom of the hypothalamus, and in the zoom of the arcuate nucleus, you can see the shape of the cell inside the precise region of interest. My images are obtained by confocal microscopy, so there are 3D images to have information of the morphology of the cell in the 3 dimension and so to analyze really the morphology. So how do I make analysis morphology? For the moment, we use Imaris software, which is a really strong software, but long and limited analysis, because I'm going to explain to you what is missing in this software. First of all the steps are manual for each image. I let you imagine I have 12 images per mice, 3 to 5 mice per diet, and it takes like 2 hours per image. So it makes a long time to have results on this analysis. The software is working that way. So first, it's making a 3D surface segmentation to isolate the cell and to create a new mask only corresponding to the cell to avoid analysis the background of the image. After, the software performs a detection algorithm, which is the filament tracer in mice, to find the somas of the cell and associate the corresponding extension that he found in the image, in the mask of the image. The problem of this software and the results is that we don't have the level of branching. There is extension getting out of the soma, and there is extension getting out of an extension. This is interesting for us, and Imaris don't give us. Then it takes 2 separate analyses to have information for the soma. So, for example, the circularity, and for extension, so the length, the number, etc. Then I told you steps are manual, so it's a non-automated system, so it takes a long time. And finally, there is no cluster system according to microglia phenotype. I explain. I have 5 or 3 diets, and we want to see maybe that a diet is gonna increase a population of microglia phenotype, or just increase the proliferation and just the ramification, but we don't have the information to make cluster, and so we don't know if there is more population of microglia in a diet or not. This is why we started the Nutrimorph algorithm, so the development of a new tool to measure morphometry of glial cell. So it's working that way. I put the same 3D image from the region of interest. Then Nutrimorph algorithm is going to perform somas detection and extension detection separately. After, it's going to connect soma to extension, to primary extension, and then primary extension to secondary extension. After this, it's going to get each object, so each cell, and get the graph extraction of each cell to get the features extraction of each cell and make clustering of the signal and of the data we have. To go a bit further on the analysis, so each step is working that way. So for the soma detection, the algorithm is going to perform first a normalization, then a thresholding, a mathematical morphology, and then it's going to make small and big elements deletion to have only cells that are real soma. And then it's going to make a soma labeling to allow us to have each soma separately, independently. After, for the extension detection, it's going to perform Frangi, vesselness enhancement, and then an histogram equalization again. After, it's going to perform thresholding and mathematical morphology, and also a small elements deletion to only have real extension, make a skeleton of this signal, and label each extension to allow then to connect each extension to the soma and other extension. For the connection part, it's going to take separately the soma and the extension, and it's going to make an iterative influence map of the glial cell based on a Dijkstra shortest path. So this tool is going to go for recording extension to the soma when information is missing by passing through the less dark voxel of the image. So to guess the signal that would be here if we had it. After, it's going to label the image, so with each soma and extension linked, and it's going to pass through the analysis part. So to analyze the signal, we perform graph extraction, as I told you, features extraction and selection. So here is an example of features we can have. For example, for the soma, it can be the volume of the soma over the volume of its convex hull. And for the extension, for example, we can have the number of extension, the type, primary or secondary. We can have the length, the thickness, the volume, etc. After getting all the features, it's going to go for a clustering. So the cluster part is in two parts. It's going to first select the features that is interesting for us. So by a feature statistical analysis and selection, and a PCR, so principal component analysis, to have an additional dimension reduction. And then it performed a clustering by using the k-means with the selected features we had to separate the data into k-clusters. So by previous work, we defined that the optimal number of k for our type of analysis is 2. So with 2 subpopulation of cell. So here is just an example of the refinement we used to do for this software, because you can see in the raw images, the problem of this type of image and this type of marker, which is IBA1, sorry. There is a lot of background in the brain. And so Nutrimorph software had a really problem to detect the extension and get it out of the background. So now with the help of Morgan Nadal, which was a master's student supervised by Eric Debreuve, we are now able to detect real extension. You can see here the brown cell have a soma which is quite comparable, and extension that are quite good. The problem still, remaining, sorry, for this software is that images are really heavy. And so it takes really a long time to make analysis and have data of it on each image. So the optimization still needed is to allow the run duration to be less long. So let's just conclude about the project. So about all I told you here, so the obesity phenotype and the weight gain is only triggered by the quantity of lipid inside the diet. So the soybean corn and the sunflower have both 20% of lipid, and they trigger the same obesity phenotype. But for the neuroinflammation and the inflammation part, only the nature of the lipids inside the diet, and so here the omega-6 and omega-3 ratio, which is really deleterious of the sunflower, triggers a strong neuroinflammation and especially a microglial reactivity. We then showed that neuroinflammation is coupled with cognitive disorders, and so only triggered here by the sunflower diet-fed mice. So this is a study really interesting because here we split obesity and neuroinflammation, because you can see that soybean corn mice are obese mice, but they don't have neuroinflammation and cognitive disorders. So what's next for the project? We have here a high-fat diet triggering neuroinflammation, obesity phenotype, and cognitive disorders associated with a strong microglial reactivity. So our hypothesis is that maybe glia and microglia can be at the origin of all these phenotypes. So what we want to do is to inhibit this neuroinflammation, and so to inhibit glia, so microglia or astrocytes, to maybe prevent or delay the obesity and avoid the cognitive disorders that are associated. To do so, we are going to use a pharmacogenetics approach, which is the DREAD technology, which consists in an injection of a specific virus for the two glia cell type I presented to you, so microglia and astrocytes, in the region of the hypothalamus. And then we're going to feed mice for 20 weeks with either the standard or the sunflower diet, and then looked at the obesity phenotype, the inflammation in peripheral and central, and the associated cognitive disorders that can be triggered. We also can do the same approach, but by injecting this virus at the end of the 20 weeks to maybe see if we reverse the obesity phenotype and the inflammation triggers. Well, I thank you for your attention all, and I would like to thank my team, Carole, Robert, Céline, Carla, and Léa, that helped me a lot with all the data. We'd like to thank Ingrid Betus and Maxime Villet for the behavioral approach on the mice, Nicolas Blondeau for this help in this project, and all the platform of my lab, so imaging, animal, and lipidomic platform, which helped me with a lot of data. I also would like to thank Zubir Amri team and Didier Pisami, which is not in the team anymore, but thank you for the project. The team and the platform of Dijon for the lipidomic analysis, and of course, my co-supervisor, Eric Debreuve, and Morgan Nadal, so the previous master student, for the help for the algorithm and the software. I would like also to thank Academy 2 and 3 of Excellence for funding in this project and implication in my master and thesis project. The IDEX, the DSK and EUR Life also, and Neuromod, of course. And again, you for your attention.
Thank you, Clara, for this terrific talk. And I think we can wait until everybody turns on the microphone, and if you can answer questions. I can start with the questions. I'd like to know if whether you use the Omero platform to do your image acquisitions.
Yes, of course. We use the Omero platform because images are really heavy, I told you. So it's better for us to transfer images from the microscope computer to mine to make analysis. And because Imari's software also is in the Omero platform, the MICA platform at IPMC. So I needed us to make this analysis on morphology.
Okay, so this means that even with the Omero platform, the Omero, applications, you need a long time to have each images.
Yes, of course. Each image takes 30 minutes in the microscope. This is the first long time of the project. And after, the problem is that images are heavy. And so we don't have a computer enough powerful to make them turn really fast. So it's working, but it takes a long time.
Okay, it's a long time. So I had another question. I wanted to know, so maybe Jean-François has a question.
Thank you, Clara, for this very nice presentation. Jean-François Tanti from C3M. I have a question regarding the inflammation in the hypothalamus. Yes. So you have any, oh, do you hear me?
Yeah.
Okay, so you have an increase in inflammation in the hypothalamus, but you have the same obesity, but do you have a change in the feeding behavior of the mice? Because there is a lot of studies showing that inflammation in the hypothalamus could be related to a change in feeding behavior.
Yes, so we measure the expression of neuropeptides as POMC, CART, MCH orexin. And for the moment, at the 20 weeks of diet, we don't have changing in the expression of these peptides. We have only the strong neuroinflammation, but I would say that expression of the neuropeptides are quite in the same phenotype as the obesity part I presented. So the soybean corn and sunflower diet fed mice do not show differences in the expression of these peptides.
Okay, despite an increase in inflammation.
Yeah, no, only the inflammation is different from the ratio.
Okay, thank you.
Jean-Louis?
Yes, great talk, my dear. I have two questions. The first one would be related to the localization of expression of this chemokine and cytokine within the brain. So do you know if there is any idea about the localization of this one? And also at the periphery, what might be the source of this cytokine and chemokine?
Well, in the periphery, we know that sometimes the source is directly the adipose tissue because increased storage make them not quite functional. And so it leads to deregulation and secretion of cytokines because there is an infiltration of immune cell inside the adipose tissue. And so a production of pro-inflammatory mediators directly inside. And for the brain, we measure in the hypothalamus. It's complicated to separate region inside the hypothalamus because it's really small, but maybe we can do, and I'm going to perform some of this analysis, RNA-scope technique, which is a, how can I say, marker of directly the cytokine and the chemokine by immunochemistry on the slice. And so maybe here we can see a different region inside the hypothalamus that could produce more cytokines and chemokines, maybe the lateral hypothalamus, or directly the arcuate nucleus, because it's the one closest to the blood-brain barrier. So I don't know, but we'll have the results soon.
We'll see within a month when you'll be back to Lille. The second question is regarding the staining. It looks like the staining for the microglia markers is stronger in the hypothalamus, in the hippocampus, sorry, when you compare with the hypothalamus. Why not starting with the hippocampus when you're doing your imaging analysis?
Of the morphology?
Yeah.
Because first in the project, we didn't want it to look at the behavior. It was not our intent. But during our experiment, I saw in the brain slice that there is a changing in the morphology in the hippocampus that I did not measure for the moment. Okay. And so we asked the question, is the behavioral is modified? And so we performed behavioral tests first because we had the mice, but it's planned. I need to make the morphology on the hippocampus then.
Okay, okay, great. Maybe a third question if I have time.
Yes, you do.
It's regarding the future. You were talking about using the DRED techniques, but there is another technique which is you'll be using a chemical component, which is the PLX 5262, if I remember well, that we use, as you know, in the glia papers. And why not starting with this one? Because it would be, I guess, complicated to interpret the result using the DRED techniques.
Yeah, so we have this chemical inhibitor. I think it's 5622. But the problem of this inhibitor is that if we want to use it, we need to let it in the food of the mice. And so it's gonna maybe trigger strong phenotype and maybe deleterious phenotype because there is 20 weeks of diet without microglia. And so maybe it's gonna be complicated for the normal functioning of the brain. But it's an approach we're gonna test, of course. The advantage, I can say, of the DRED technique is that it can be activated in the moment we want. So we can activate the DRED technique at the very beginning, or at 4 weeks, at the really beginning of the obesity, or at the end, after 20 weeks, just to see if we reverse something.
Okay. Okay, thanks a lot.
Okay, thank you. There's a question from S. Zubir Amri. He wants to know, did you give a look, if you did you give a look to the lipid composition of membranes in hypothalamus?
No, not for the moment. We wanted to, but the problem is that our lipidomic platform is not able to measure the free fatty acid inside a tissue or the serum of mice. So it implicates us to make a new batch of mice and to have the hypothalamus to send to another platform. And for the moment, we didn't have the time. But of course, it's really interesting and we want to do it, yes.
Okay, thank you. I wanted to know if you have an effect of lipids on other part of the brain or other cell types other than microglia.
So I'm going to perform the same analysis for the astrocytes, as I told you, because there are two glial cell type and really related here in the neuroinflammation. So I'm going to make a morphologic analysis of the astrocyte too. And for the specific region, I want to, but for the moment I don't have the time, I would like to cartography, I don't know if it's the word, but
haptocartography?
Yeah, of the whole brain slice to see if there are some region with a strong marker of IBA1 or GFAP for the astrocytes, maybe to see if there is another region of the brain other than the hypothalamus and hippocampus implicated in the neuroinflammation here.
Okay. Okay. Any other questions? No, I think, let me, no, maybe Jean-Louis? No, he still has,
Jean-François.
Jean-François.
Yes, maybe another question. Do you know if Do you know if the neuroinflammation is an early event during the development of the obesity in your model? Did you check at different time after the beginning of the diet?
Yeah. So it's a study we recently published in Glia that we test in the lab, that after 1 hour, 3 hours, or 6 hours of diet, there is neuroinflammation starting in the brain. And so here is I don't know if it's a bit messy, but you can see that we perform a first analysis with the Nutrimorph algorithm, I told you, and we obtained an increase in the proportion of the glial cell at 3 hours of diet. So this is really showing that there is a neuroinflammation here at 3 hours of diet. For my part, and maybe more long diets, we measure at 4 weeks, 8 weeks, 12 weeks, and 20 weeks. I presented you the 20 weeks. We start to have neuroinflammation really strongly here at 8, really 12. At 4, we don't see nothing.
Okay. And both in the hypothalamus and the hippocampus? And,
hypothalamus, yes. Hippocampus, I did not test it for the moment at the early time.
At the early time. Okay, thank you.
Isabelle?
Yes. Do you hear me?
Yes, we cannot see you.
Yes. Here I Hello. Thank you very much, Clara. It was very clear and interesting. Maybe I missed it, but I was wondering, is there any studies that that would predict or say that people with obese people are more what you said, there is some symptoms of anxiety and also of hyperactivity and so on.
For the cognitive disorders?
Yeah, some cognitive disorders.
Yeah, there are studies showing that obese people are more tending to go for depression and anxiety behavior in humans. That is why we tested it in mice. For the hyperactive part, I don't know. I'm not sure. Maybe not. But it was interesting in mice because it's a behavior related and anxiety-like behavior in mice. And so it was the precursor of our analysis for behavior.
Mm-hmm. So it would be very interesting to know if there is a link, maybe for young people, for children, a link between obesity and hyperactivity, for example.
Yeah, maybe.
Thank you very much.
Amri has another question. He wants to know, did you try to revert the effects? Put the mice again on show diet after SF diets?
So no, we did not revert for the moment, but I know that it's been done in a study, I don't remember, where they tried to reverse after 4 weeks of high-fat diet, and they do reverse a bit the weight gain. But I think that after 8 or 12, it's not possible to revert totally the weight gain of the mice. But we did not do it here in my studies.
Okay.
And I can see Nadine, maybe a question.
Yeah, did you work only with male mice?
So yes, for the moment, we only worked with male mice because I had really a lot of mice in each time, because I told you in a question, I had the time at 4 weeks, 8 weeks, 12 weeks. Each time represent like 30 or 50 mice. And so we didn't have the place first and my time secondly to have female in the same time. But it's going to be interesting. In another time, for the Glia paper we published, we measured all the data on male and female, and we didn't have a difference between the two sexes. So it's going to be interesting to do it for my experiment, so with long-term high-fat diet. But we have an information that at early time, it's not different between the male and female mice.
Okay. No more questions? No?
Yeah.
I wanted to thank you all for attending, for your question. Thank you, Clara, for this very good presentation. And I give you rendezvous on the next Academy4 Research webinar. So it's going to be the 15th of June, so next month. And we're going to talk about immunity and sports. So see you next month and take care. Bye-bye.
Thank you. Bye.
Thank you. Bye.